Hi everyone, hope you're well. Welcome to the session. This is the latest lecture in MD 6028. So today we're going to talk specifically about viral oncogenesis. So like last week, we're looking at a kind of niche subsection within this idea of viral pathogenesis. So within the idea that viruses cause disease in certain ways. This is the portion of the lecture which we refer to as viruses as agents of disease or viruses as human pathogens. So again, we're going into some depth looking at how viruses specifically cause disease, and viral oncogenesis is one of those areas kind of within this subtopic that is so different from how viruses normally cause disease. It's so interesting and it's so unique that it really needs to be studied a little bit separately, similar to the bacteriophages last week. It just needs to have its own space, and we need to think about it in a little bit of depth. So that's what we're going to do today. We're going to look at viral oncogenesis, and what you're going to see is that the process of viral oncogenesis is again very different to how viruses normally cause disease. Over the course of this module and in other modules, you've looked at a range of different diseases that viruses cause. You'll see that in the case of viral oncogenesis, things are just a little bit different. The principles are still the same in the sense that the rules still apply. It's just there's some unique and strange things going on. And that's what we're going to consider today. So in some past lectures, we have touched on this idea. I didn't really go into any depth. I kind of brushed it aside a little bit and said, oh, we're going to talk about this more later. Later is now, basically. So this is the lecture in which we'll go into viral oncogenesis in some level of depth. So to discuss viral oncogenesis, we need to understand what cancer is, really. We need to understand this process. We don't need to understand cancer or the process of cancer formation in a huge amount of depth. We just need to understand it well enough to allow us to progress through this lecture and understand the part that viruses can play. So when we're talking about cancer, what we're essentially talking about is uncontrolled proliferation of cells. So we're talking about uncontrolled growth. This is probably familiar to you, that this is what cancer is. It's uncontrolled growth. It's hyper proliferation, hyper meaning over. And this hyper proliferation is what leads to the formation of tumors. So it's what leads to tumorigenesis, which is again an aspect of and quite synonymous with the idea of oncogenesis. So the idea of cancer formation overall. That's what a tumor is essentially. It's a hyperproliferation. It's an overgrowth of cells. It's your own cells that have grown in an uncontrolled manner into a tumor. We make this distinction, and we've made this distinction before, between transformation and oncogenesis. And this distinction is really, really important because one flows into the other. So transformation is a prerequisite for oncogenesis or tumorigenesis, and transformation is a cellular event that occurs before the hyperproliferation. Okay, so normally, and we're going to look at this in the next few slides, cells have a really, really controlled manner of replication. So they progress through the cell cycle in a very, very controlled way. There's very specific checkpoints that we'll look at. However, when cells are hyperproliferating, so when they're on their way towards potentially forming a tumor, they are not replicating in a controlled way. Again, that's why we say hyperproliferation. The cell cycle has been what we call dysregulated. Now, the process of how a cell gets from replicating in a healthy way to hyperproliferating is referred to as transformation. And this process, transformation, is potentially caused by viruses. Obviously, it's not always caused by viruses because we have many instances of cancers that aren't linked to viral infection. However, some viruses are capable of causing transformation, and this transformation can lead to hyperproliferation, which can lead to tumorigenesis it's really important that we understand this distinction between transformation and kind of tumorigenesis and oncogenesis. Transformation is the prerequisite. Transformation is the cellular process by which a cell goes from replicating in a healthy manner to hyperproliferating. And then once this state of hyperproliferation is occurring, then there can be progression to oncogenesis and tumorigenesis. I've outlined the term here, tumorigenic viruses. You'll see the term tumorigenic viruses or oncogenic viruses used interchangeably, really, and that's absolutely fine. They're just referring to viruses that can cause this process to occur. And again, what these viruses are specifically causing is transformation. They are altering the cell in such a way that they are dysregulating cell cycle progression, and they're causing the cell to therefore replicate in an uncontrolled manner, which we define as hyperproliferation. And this is the cell cycle I've been talking about. Again, I'm sure this is familiar to you. We've seen it in this module, so it is familiar to you. But in your cell biology modules in the past and things like that, I'm sure you've talked about the cell cycle. What we need to know in the context of this lecture is that this is a cyclical process. So cells progress through this cycle in a clockwise manner. So from M phase to G1 phase to S phase to G2 phase, they progress through these phases, and each progression around this cycle results in cell division. That's why we call it the cell cycle. And that's why it's a cycle, because you can get progression over and over and over again. So you get multiple rounds of division. However, that is usually in a healthy cell controlled. So progression through this cycle in a healthy cell is very, very tightly controlled. It's not just whizzing around, constantly dividing, going from stage to stage to stage to stage. That's not how it works. There is a very slow, controlled progression through this cycle. And these two checkpoints are crucial in this process. So we have the G1S checkpoint and we have the G2M checkpoint. These checkpoints are instances in the cell cycle where there are checks, there are checkpoint, where there are cellular mechanisms to make sure the cell is ready to progress to the next phase. So to make sure the cell is ready to progress to either S phase for the G1S checkpoint or to M phase for the G2M checkpoint. So progression through the cell cycle is essentially stalled at each of these two checkpoints until the cell is ready to progress to the next phase. Obviously, we're not talking about a conscious decision here. We're talking about molecular mechanisms that are monitoring the cell's status and monitoring whether the cell is ready to pass through to the next phase. So we're talking about cellular mechanisms, and again, the purpose of these cellular mechanisms is to ensure the cell is prepared to move to the next phase. Oncogenic viruses. So oncogenic viruses, as we've said, will transform cells, or they won't always transform cells, but they can transform cells. That's how they induce oncogenesis. They transform the cells, and then this progression towards oncogenesis, tumorigenesis can occur. So that's the principle. What we want to do now is discuss some of these oncogenic viruses specifically, and it makes sense to start at the beginning. So we're going to go right back to the early 1900s, and we're going to talk about a doctor called Peyton Roos. Now, you might have heard of the Roos sarcoma virus. The Roos sarcoma virus was named after Dr. Peyton Roos. Now, he first discovered a virus, which it turned out was the Roos sarcoma virus, but it hadn't been named yet. So he would eventually name it the Roos sarcoma virus, we'll say. He discovered it as a filterable agent. The term virus wasn't really used then, it wasn't established, so he identified it as a filterable agent. and he discovered that this filterable agent can be used to induce tumours in healthy chickens. So he took tumours from English hens, he isolated something from these tumours that he just defined as a filterable agent, and then he used this agent to induce tumours in healthy chickens. So straight away, this is proof of principle that this agent, whatever it is capable of inducing tumorigenesis. So what we're looking at here is cases of cancer that are caused by an infectious agent, caused by something that can migrate from host to host. Obviously, it's being mediated by the experiment, but the principle is still there. There's a causative agent that is present in the original tumours, and that can induce tumours in the healthy chickens. And what was actually happening here, as we now know, was there was an oncogenic virus. So this agent that was being isolated was an oncogenic virus, and it was in fact a retrovirus, the first retrovirus discovered. What we're going to see is retroviruses crop up over and over again in this lecture. So a lot of oncogenic viruses are retroviruses, or we can say a lot of retroviruses are oncogenic. Why is this the case? It will become clear as we progress through the lecture. I can give you a broad idea now and say that one of the ways, or the principal way in which oncogenic viruses transform cells is by influencing their genome, so inducing changes in their genome. One thing we know about retroviruses is they are capable of integrating their own genome into the host cell genome, so it shouldn't really be a surprise that these retroviruses are often capable of inducing transformation, because we already know they have the tools to induce changes in the host cell genome, and that main change is in integrating their own genome into the host cell genome. What we have here is a table of the different retroviruses. So as you said, the retroviruses are part of a family, Retroviridae, and if we remember the suffix viridae means family, that's what we're talking about. Within the Retroviridae family, we have these different genuses of the retroviruses. We have a number here, and then we have the individual species. So this is quite a useful table to put things in perspective. So it's quite useful overall, but when we're talking about oncogenic viruses, it's particularly useful because we can see the names of the different species. And what you should notice is a lot of the names indicate the ability of these retroviruses to cause tumours. So they indicate at the oncogenic nature of these virus. And so we have Rous sarcoma virus. What else have we got? Nasal tumour virus, leukemia virus, murine leukemia virus, woolly monkey sarcoma virus, bovine leukemia virus, epidermal hyperplasia virus. So it's pretty clear that there's a common theme in the naming of a lot of these retroviruses, and this common theme is linked to the ability of these retroviruses to induce transformation, which down the line results in hyperplasia or hyperproliferation and tumorigenesis and oncogenesis. And again, what we're going to do in this lecture is try to understand why so many of these retroviruses are capable of inducing these effects. But I think this is quite a useful slide because it is quite startling that so many different retroviruses have been named essentially for their ability to induce oncogenesis. Okay, so we've gone through some of the foundational principles of oncogenesis and viral oncogenesis specifically. What we want to do now is look at the two kind of subgroups of oncogenic viruses. So we want to think about oncogenic RNA viruses and also think about oncogenic DNA viruses, and think about the different way these two groups of oncogenic viruses can actually transform cells, so the different way in which they actually cause oncogenesis. Now, I've introduced this idea of oncogenic retroviruses, so the idea that a lot of retroviruses are oncogenic. Obviously, retroviruses are RNA viruses, which I'm sure you know. So this first section, we're going to be talking about oncogenic RNA viruses, and retroviruses fall into this category. So the ability of RNA viruses to induce oncogenesis is revolving around what we call oncogenes. So oncogenes are essentially genes that are directly responsible for transformation. That's hence the name onco, meaning cancer, and we know transformation is a precursor to the formation of tumors, and so for tumorigenesis and oncogenesis, and gene because it's a gene. So you've probably heard this term oncogene. You probably know what it means, to be honest. You've probably covered it in cell biology modules, but what we're going to do now is think about these oncogenes in the context of viral infection, specifically in the context of oncogenic viruses. So we can think of oncogenes as being pro-transformation, so they induce transformation. Now, the role of viruses in this process is they can either activate oncogenes that are already in the cell, or they can introduce new oncogenes into the cell. So two strategies there. Both of these strategies end with a cell expressing oncogenes that it wasn't expressing before. So whether the virus brings the oncogene into the cell, or the virus activates oncogenes within the cell that weren't necessarily being activated before, so that weren't necessarily being expressed before. By either of these strategies, some viruses can induce transformation by inducing the expression of these oncogenes. So again, whether they're bringing the oncogenes in or they're activating the oncogenes already there, they're promoting the expression of oncogenes within the cell. And again, when we're using this term oncogene, we're referring to genes that when expressed, cause cellular changes that contribute to transformation. So they transform the cell. There's a bit of terminology here where we need to think about V oncogenes and C oncogenes. The V and C, we can just think of them as meaning viral or cellular. So we have viral oncogenes and we have cellular oncogenes. So the viral oncogenes are oncogenes that are present in the viral genome, and these are the oncogenes that viruses introduce into the cell. So if we have a virus with an oncogene in its genome, this virus infects the cell, then it's potentially what it's doing, assuming that gene is expressed, etc., potentially what it's doing is transforming the cell, because it's bringing in an oncogene that can then potentially be expressed. And we'll see there's some mechanistic things involved and it's a little bit more complicated, but that's the principle. The virus is introducing a viral oncogene into the cell, and those are V oncogenes. C oncogenes, on the other hand, these are cellular oncogenes. So these are oncogenes that are present within the cell. They are part of the cell's normal genome, but some viruses, when they come in, are potentially able to activate these oncogenes. So they're potentially able to activate C oncogenes, cellular oncogenes, whether it's a V oncogene or a C oncogene. So whether it's the virus bringing the oncogene in or the virus activating a cellular oncogene, and we can see that the virus is potentially contributing to transformation. And a few slides ago, we discussed how retroviruses can very often cause transformation of cells, leading to oncogenesis. So we looked at all the examples of different retroviruses that are oncogenic. We only really looked at their names, but still we kind of demonstrated that principle. This is why. So the two strategies here, or the two mechanisms by which transformation can be induced, the introduction of oncogenes directly or the activation of cellular oncogenes. Both of these mechanisms can occur as a result of retroviral genome integration into the host cell genome. So when a retrovirus introduces its own genome into the host cell genome, when it integrates that genome, it can potentially be integrating an oncogene as part of its own genome. So if it has an oncogene in its genome, when it integrates its genome into the host cell genome, it will of course also be integrating that oncogene. And that is mechanism A listed here. It's introducing an oncogene into the host cell genome. The other mechanism, as we said, is that activation of host cell oncogenes. So viruses can potentially activate oncogenes that are already present in the cell. What we're going to look at in a little bit is the fact that when a retrovirus introduces its own genome into the host cell genome, so when it integrates its genome into the host cell genome, it can potentially activate nearby oncogenes that are already in the host cell genome. So this mechanism is an example of how a retrovirus can activate oncogenes that are already there. Retroviruses are therefore potentially capable of inducing cellular transformation by these two mechanisms, and we do see this. We see retroviruses inducing transformation by these two mechanisms. If we compare retroviruses to non-retroviruses, so to viruses that don't integrate their genome into the host cell genome, it becomes pretty clear how and why a large number of retroviruses are oncogenic. Again, by integrating their own genome into the host cell genome, they are the opportunity to induce transformation is there. The opportunity to introduce new oncogenes into the host cell genome, or to induce nearby oncogenes already present in the host cell genome, presents itself because retroviruses are inherently manipulating the host cell genome because they're inserting their own genome into the host cell genome. So again, because of this process of integration, it shouldn't be a big surprise that so many retroviruses are capable of inducing cellular transformation. Okay, so these oncogenic viruses, there's a little bit more to say because we can actually subdivide them into three groups. We can subdivide them into the transducing viruses, the non-transducing viruses, and the non-transducing long latency viruses. And what we see when we explore these terms is that they align very, very well with the mechanisms we looked at just a couple of slides ago. So with the ability of some viruses to introduce new oncogenes into the cell, and the ability of other viruses to activate oncogenes that are already in the cell. So for example, these transducing viruses, you'll sometimes see them referred to as high efficiency because they induce tumors with high efficiency. It's almost 100% of the time infection results in transformation and then down the line tumorigenesis. So very high efficiency. The reason why they are high efficiency is because they utilize oncogenes that they carry into the cell. So the virus has an oncogene in its genome, it infects the cell, and it's bringing this oncogene in. Again, we refer to these oncogenes as V-oncogenes because they're viral oncogenes because they're being brought in by the virus. And again, these are transducing viruses and they are high efficiency, up there near 100% in terms of the frequency of transformation. You'll see the term cell-derived oncogene used, and that's another way we refer to these V-oncogenes. So another way we refer to these viral oncogenes. It can seem a little bit strange because the other type of oncogene is what we refer to as a cell oncogene. Cell-derived is not the same as cell. Okay, so V-oncogenes are viral oncogenes and they're cell-derived. What that means is the virus originally got that oncogene from a cell. So the way these viruses that carry their own oncogenes obtain these oncogenes, the way they get them in the first place, is they steal them from cells. They acquire them from cells. That's why we call these viral oncogenes cell-derived oncogenes sometimes. Now, the non-transducing viruses, these have varying efficiency. They're nowhere near as efficient as the transducing viruses are inducing transformation. This is because they don't carry their own oncogenes. They don't induce via viral oncogenes. They activate cellular oncogenes. You'll see this term proto-oncogenes sometimes, because sometimes we'll refer to oncogenes as proto-oncogenes before they're activated, because even though they have the capacity to be an oncogene, even if they're expressed, they will cause transformation. We refer to them as proto-oncogenes. There's a little bit of more technical information we need to talk about this, because these oncogenes, or these proto-oncogenes, often, if they're expressed at a normal level, they don't cause transformation, so they can't really be considered oncogenes because they're expressed normally within the cell and they don't cause transformation. However, the introduction of a non-transducing virus can activate this gene, and it causes overexpression of this gene, and when this gene is overexpressed, it then potentially induces transformation. That is why we can refer to some of these oncogenes as proto-oncogenes, because normally they're just a normal gene. If they're expressed at normal level, we can't really consider them an oncogene because they're just they're normal. They're not causing oncogenesis. They're not causing transformation. However, if they are overactivated by non-transducing viruses, then they function as an oncogene, then they can induce transformation. So we tend to refer to these oncogenes as proto-oncogenes until they're activated by a non-transducing virus. There's actually two forms of activation. There's C-activation and there's trans-activation. These non-transducing viruses are C-activating. We also have non-transducing long latency viruses, which are low efficiency, very low efficiency. And the reason why they're low efficiency is because they are trans-activating. So like the non-transducing viruses, these long latency non-transducing viruses, they don't bring their own oncogene into the cell. They don't have viral oncogenes. They don't have cell-derived oncogenes, which is another way of referring to these viral oncogenes. They actually activate host cell oncogenes, just like the other non-transducing viruses. However, these long latency non-transducing viruses activate these cellular oncogenes in a slightly different way. They activate them in trans, so we refer to them as trans-activating, and I'm going to go into this in a little bit more detail in a second. Okay, as we've said, transducing viruses, these are introducing viral oncogenes into the host genome, whereas non-transducing viruses and the non-transducing long latency viruses are activating C oncogenes that are already in the host genome. So if we now quickly explore this distinction between C activation and trans activation. So the non-transducing viruses activate via C activation. What this essentially means is they will activate oncogenes that are physically next to their position of integration. So when they integrate next to an oncogene, they can activate the oncogene by virtue of being close to it in the genome. That's all that C activation means. Transactivation, which the long latency viruses engage with. Transactivation means the activation of oncogenes that are in a separate region of the genome, so they're not necessarily physically close to the site of integration. So we're looking at two slightly different mechanisms by which the cellular oncogenes are activated. And again, we see different efficiency. We see different efficiency within the non-transducing viruses, but with the non-transducing long latency viruses, what we see is generally low efficiency. So we can say there's a difference in efficiency generally between the non-transducing viruses and the non-transducing long latency viruses. And it's fundamentally because of this difference in mechanism of oncogene activation. So the difference between C activation and trans activation. Okay, this is kind of a little bit more information in terms of the idea of viral oncogenes. So I just inferred, and I kind of just slipped it in without real explanation, that these viral oncogenes can sometimes be referred to as cell-derived oncogenes, and that implies that the place these viruses originally got their viral oncogene from is a cell, because it's cell-derived, and that's absolutely the case. These viral oncogenes originated as cellular oncogenes that viruses acquired. So they obtained the oncogene from cells, and then it's then referred to as a viral oncogene or as a cell-derived oncogene. We used the Wu-sarcoma virus as an example earlier, and we can use that example again essentially because the Wu-sarcoma virus, it carries a viral oncogene, it carries VSRC, so viral SRC, and this viral SRC is derived from cellular SRC. It's the first oncogene discovered, and it encodes a tyrosine kinase. And what this tyrosine kinase ultimately does is it stimulates uncontrolled cell replication. It stimulates mitosis. This isn't unusual. So what we see when we look at these oncogenes' mechanisms of action is they promote cell division. You know, we kind of should expect that, shouldn't we? Because these oncogenes are pro-proliferative. They are contributing or directly causing cellular transformation, and as we've said, cellular transformation is hyperproliferation. So again, it shouldn't be a surprise that these oncogenes often have a role in promoting cell division, and that's clearly the case for SRC. The question then becomes, okay, how did Wu-sarcoma virus gain SRC, or how did any of these oncogenic RNA viruses obtain the viral oncogene? How did they derive it from a cell gene? How did they get it? And the answer really is that we're not 100% sure. So we have some ideas of potential mechanisms which we'll talk through. It's difficult to know for sure. The mechanism we'll look at next is really reasonable, and it's quite possible this is correct. Again, we're trying to figure out something that's already happened. We're trying to infer how it might have happened, which is, you know, it can be quite difficult to do things like that. But we'll look at this mechanism and see what you think. This graphic should be fairly familiar to you because it's something we've talked about before at length. It's the retroviral process of reverse transcription and genome insertion into the host cell genome. So you can see we're starting off with the viral RNA genome at the top in light blue. This is reverse transcribed into a DNA copy, and this DNA copy is then inserted into the host cell genome. When this DNA copy is inserted into the host cell genome, it can then be used for genome expression, and it can also be used to produce the RNA copies of the genome that are required for they represent genome replication, right? Now, what we want to think about is how this aligns with the idea of retroviruses capturing oncogenes, and we'll talk through it and hopefully it will be clear. Now, again, from past modules you've done, I'm really sure you're aware of what stop codons are, but as a super quick recap, stop codon is a sequence in the genetic information, so in the genome, that signals the polymerase to stop. So the RNA polymerase in this case that is progressing along the provirus to stop transcribing. There's a stop signal at the end of the viral genome, and the purpose of this stop signal is so that the viral genome isn't including a whole bunch of host genome information on the end. So if there's no stop codon at the end of the viral genome, the RNA polymerase is going to keep on going, and then the full length copies of the retroviral genome will have a bunch of junk at the end, will have a bunch of host genome information included, like by accident really. The virus obviously doesn't want this, so the virus has stop codons included at the end of the genome. These are included in the what we call the LTR, which is the long terminal repeat region. So this is a region that doesn't have the information that doesn't encode proteins. It's just a lot of junk, a lot of repeats, long terminal repeats. It's partially to protect the genome from damage, because if the ends get kind of snipped off or the ends get degraded, it doesn't actually cut into the genetic information that's encoding proteins. It's just cutting into these long terminal repeats, and as part of these long terminal repeats there is a stop codon that tells the RNA polymerase to not progress any further. So it's a signal that this is the end of the genome that needs to be expressed. The issue is that occasionally this stop codon can get altered so that it's not sufficient anymore, so that it doesn't effectively signal, if you like, the RNA polymerase to stop. If there's a mutation in the stop codon, then the RNA polymerase may no longer recognize that stop codon, and it will continue progressing and it will continue transcribing, so you end up with a viral genome that has extra information on the end. This extra information could potentially include genes that encode oncogenes that are then becoming viral oncogenes because they're being included in the retroviral genome. So this is one theory as to the origin of these viral oncogenes. So they start off as cellular oncogenes. The virus has happened to integrate its genome right next to one of these cellular oncogenes. When the RNA polymerase is expressing the viral genome, so producing an RNA copy of the viral genome that will then be processed into the viral particle, so that will serve as the virus's genome, this RNA polymerase has come to the stop codon. The stop codon, however, is mutated, and the RNA polymerase has continued past the stop codon and has included the cellular oncogene in the viral genome. And this cellular oncogene, now it's included in the viral genome, is referred to as a cell-derived oncogene or a viral oncogene. And this makes sense. I mean, there's a couple of things happening that sound a little bit unlikely. So for this to work, first off, you need the mutation in the stop codon. Also, you need to have the retroviral genome incorporated just upstream of the cellular oncogene. And yeah, these things are fairly unlikely to happen together. However, we're talking about evolutionary history. We're talking about a long period of time, right? And statistically, there's no reason that this couldn't have happened. There's absolutely no reason. So it does actually make sense, even though these things sound unlikely. If you think about the number of these viral infections that are occurring over a long period of time, it almost seems inevitable that this would eventually happen, that you'd eventually get integration of the viral genome just upstream of an oncogene, and then you would also happen to get a mutation of the stop codon. That would mean the RNA polymerase progresses past the end of the viral genome and includes an oncogene into the viral genome. And again, this is how the viral oncogenes may have originated. Okay, so now we want to think about something called the kinetics of oncogenesis. We talked about the efficiency of oncogenesis when we looked at the different types of oncogenic viruses. We said some are high efficiency, causing tumors close to 100% of the time. Some are low efficiency, so the non-transducing viruses are varying efficiency, but they're lower efficiency than the transducing viruses, right? And the latent ones, the long latency non-transducing viruses in particular, these are very, very low efficiency. And again, that's because they're activating cellular oncogenes in trans. We can also now think about the kinetics of tumor formation. So efficiency is how how efficient the virus is at inducing tumorigenesis. So how likely any given infection is to induce tumorigenesis, if you like. Kinetics is to do with how fast tumor formation occurs. So how fast does this oncogenesis take place? Is it rapid or is it slow? And then in the middle we have this classification, intermediate. These classifications align very well with the classifications we looked at a few slides ago, and you might be expecting this. What we see is the high efficiency viruses, and again, these are the transducing viruses that carry viral oncogenes. These induce oncogenesis in a very, very rapid manner, so we refer to these as being rapid. Again, fast. They're very quick to induce these tumors. The intermediate viruses, so these are the viruses that induce oncogenesis or tumorigenesis in an intermediate manner. These don't carry their own oncogene, however, they activate cellular oncogenes in C manner, so it's C activation or cis activation. If you go back a few slides, you remember what I mean by that. These are the non-transducing viruses. They're activating cellular oncogenes by inserting near to those oncogenes, and by virtue of being close to them, they're activating their expression. Then we have the oncogenic viruses that have a slow, the kinetics of oncogenesis is slow, so they don't rapidly induce tumorigenesis, they do so very slowly. These are in line with the third group we looked at a few slides ago. So these are non-transducing viruses that are long latency. And again, that term long latency hints at this ability to slowly induce tumorigenesis, to induce tumorigenesis after this significant delay or over this long period of time. And again, the reason is because they are trans-activating the cellular oncogenes. So they don't carry their own viral oncogenes, they activate cellular oncogenes, and they do so by trans-activation. Okay, and this is kind of aligning things to what we looked at a few slides ago. So again, transducing viruses, rapid oncogenesis, non-transducing viruses, intermediate oncogenesis, and the non-transducing latent viruses, we have slow oncogenesis. Okay, so those are the oncogenic RNA viruses. Quite a bit of technical information there, but I hope it was effectively explained. Hope it's clear. Now we want to think about the oncogenic DNA viruses. So with the oncogenic RNA viruses, we focused a lot on retroviruses because that's inherently the focus of the area. These retroviruses are really, really important. With DNA viruses, we're going to move away from retroviruses because retroviruses are RNA viruses. A number of DNA viruses can cause tumorigenesis, so a number can be considered oncogenic. The mechanisms of oncogenesis are different to what we see with the RNA viruses, and hopefully it'll be clear what I mean by that. Okay, so we're probably very, very familiar with the papillomaviruses and with the idea that human papillomaviruses can cause oncogenesis. We've talked about this at length, right? We've talked about how HPV can cause hyperproliferation, and that can lead to cervical cancer, it can lead to anal cancers, it can lead to oropharyngeal cancers. Wherever the site of HPV infection is, it can potentially be causing hyperproliferation. And again, that hyperproliferation is a result of transformation. So these DNA viruses, like the RNA viruses, are oncogenic because they're inducing transformation. Again, the mechanism is different though. With these DNA viruses, the oncogenic DNA viruses, there's a strong focus on what we call T antigens. Now, T antigens are specific proteins carried by these viruses, and these proteins play a function in the transformation of the infected cells. Now, when we're referring to T antigens, specifically we're referring to polyomaviruses and papillomaviruses. However, we'll also quite often use T antigens as a general term to refer to the proteins that DNA viruses encode that cause transformation. So to refer to the oncogenes that are carried by these DNA viruses, or the oncoproteins that are encoded by the oncogenes of these DNA viruses. Now, these T antigens are essentially carried by the viruses. So some DNA viruses carry these T antigens, and these DNA viruses can often cause tumorigenesis. And again, it's the T antigen that is actually actively doing this. The viruses that carry these T antigens need these T antigens. The T antigens enable their replication, but they also cause transformation. And we're going to look specifically at the way in which the T antigens can do this. When we think about how these T antigens induce transformation, we need to think more generally about how oncogenes induce transformation, because these T antigens are proteins and they're encoded for by genes, which we can refer to as oncogenes really. The oncogenes we've already discussed, so that are encoded by the RNA viruses, these also encode proteins that induce transformation. And we've kind of brushed over this point so far. We've discussed the fact that oncogenes, when expressed, induce transformation, but we haven't actually said how. We haven't said how the proteins that these oncogenes encode induce transformation of the cell. How do they cause specific changes that cause the cell to hyperproliferate? And that's what we're going to do now. We're going to think about the proteins that the oncogenes encode and how they induce hyperproliferation. We're going to think about the T antigens carried by the DNA viruses and how these induce transformation. So these are three of the questions that we've kind of answered two of, right? We've discussed how viral oncogenes are introduced. We've introduced how C oncogenes are activated, but we haven't actually discussed how these oncogenes cause transformation. So that's what we want to do now. And again, this how do oncogenes cause transformation question also applies to the T antigens. So whether we're thinking about RNA viruses or DNA viruses, we're talking about the use of genes that encode specific proteins that in turn induce transformation of cells. So again, how is this done? There's really two principal mechanisms. One is by impacting cellular signaling pathways. So there's various cellular signaling pathways that exert control over cell cycle progression. The other way is to directly dysregulate the cell cycle by manipulating checkpoints. Earlier in the lecture, we discussed in some degree of depth the idea that cells progress through the cell cycle in a controlled manner, and this control is exerted through cell cycle checkpoints. So there are two specific checkpoints, the G2M checkpoint and the G1S checkpoint, that the cell must progress through to replicate. So these are checkpoints in the progression cycle, and the cell can be stalled at these checkpoints if, for various reasons, it's determined by molecular mechanisms that the cell is not ready to progress through the cell cycle. So again, these are control checkpoints, and certain oncogenes, when expressed, can dysregulate the cell cycle, again, by manipulating these cell cycle checkpoints. So, firstly, we want to discuss oncogenes impacting cell signaling pathways. One thing we have to understand here is that cell progression through the cell cycle doesn't occur randomly, really. It's often induced in response to specific cell signaling events. So there will be signaling events that induce replication of the cell, that essentially tell the cell that replication can occur, that the environment is favorable for replication. So the signaling events, these signaling events signal for the cell to pass through the cell cycle. So they're signaling for the cell to pass through the cell cycle checkpoints. Certain oncogenes, when expressed, will manipulate this process. So certain oncogenes encode proteins that manipulate this cell signaling. And SRC is an example of this. So SRC in its cellular form is a protein that promotes cell cycle progression. And again, this is normal. There's nothing abnormal about a protein that promotes cell cycle progression, because this is a regulated process. So there'll be a push and pull in both directions. There'll be signals that are telling the cell to go through the cell cycle. There'll be other signals telling it not to go through the cell cycle, and these signals will depend upon what extracellular signals are coming into the cell. This is all part of how the cell controls progression through the cell cycle. The problem, however, is that viral SRC is going to be constitutively active, right? When the virus brings this SRC into the cell, you're going to end up with large amounts of this SRC being produced, this viral SRC, and this SRC is going to be inducing proliferation of the cell. It's going to be inducing hyperproliferation. This viral SRC, and this is crucial, is different from cellular SRC. So cellular SRC is controllable. It can be switched on and it can be switched off, and this is one of the reasons why control through the cell cycle is so it's one of the reasons why progression through the cell cycle is controlled. There isn't this constitutive progression. It's not constant whizzing around the cell cycle. There is a degree of control, and one level of control is where the SRC is switched on or switched off. With viral SRC, it is constitutively switched on. It is always active, and therefore it is, by virtue of always being active, it is activating other pathways in the cell that promote cell cycle progression. So in a normal healthy cell, SRC isn't always active, so it's not always promoting cell cycle progression. It's not always promoting those signaling pathways. However, viral SRC is constitutively active. So when a virus introduces viral SRC into the cell, that viral SRC constantly induces cell signaling events that promote proliferation, and it's therefore not surprising that viral SRC induces cellular transformation. As we discussed earlier, not all of these viruses that induce oncogenesis carry oncogenes. Some of them will activate cellular oncogenes within the cell. And again, this is often insertional activation, where the virus will insert its genome next to a cellular oncogene, and in doing so will activate that cellular oncogene. One example of this is MYC expression by virus-associated viruses 1 and 2. So MYC is it works in a similar way to SRC in principle. So it's promoting cell signaling events that nudge the cell towards proliferation, so pushes the cell towards hyperproliferation. And virus-associated viruses 1 and 2 are essentially insertionally activating MYC. So once you get this infection, you get overexpression of MYC, so the cell moves towards a proliferative phenotype, so it moves towards hyperproliferation. And that is how these viruses induce B cell tumors. Again, there's no new form of the oncogene being introduced. There's no constitutively active form of the oncogene being introduced, which is what we looked at in the last slide. That's not happening here. What is happening here is the cellular oncogene, the cellular version of the gene, is being expressed at an increased level, and that is happening as a direct result of the viral infection. Next, we have oncogenes that dysregulate cell cycle checkpoints directly. So these aren't manipulating the cell signaling events that are going on upstream that are promoting cell cycling. These are instead directly interacting with proteins that, again, directly control the cell cycle checkpoints. So they are having this direct impact on the cell cycle itself. Now, the T antigens that we introduced as belonging to some DNA viruses are actually really good examples of proteins that do this. So T antigens, for example, will interact with retinoblastoma, and retinoblastoma is a protein that typically prevents progression through the cell cycle. So we have G1S checkpoint labeled here. Now it's the progression point from the G1 phase to the S phase. It's the checkpoint fundamentally. Retinoblastoma, RB, is a cellular protein that protects the integrity of this checkpoint. It prevents the cell from progressing through the checkpoint. So again, it's controlling, it's preventing uncontrolled proliferation. One thing T antigens do is inactivate retinoblastoma, so they bind to retinoblastoma and prevent it from fulfilling its function. You may have heard of retinoblastoma as being referred to as a tumor suppressor gene, and this is why we refer to retinoblastoma as a tumor suppressor gene, because it's preventing the process of transformation because it's controlling the checkpoint from G1 phase to S phase. Therefore, it's understandable that when T antigens overcome retinoblastoma, they push the cell towards transformation because they're fundamentally promoting proliferation. So let's explore a little bit about what's happening with retinoblastoma. So in a pro-replicative situation, so signaling is coming down into the cell that is promoting replication. It's triggering mitosis. What you see with retinoblastoma is it's phosphorylated. This phosphorylation essentially inactivates retinoblastoma, because normally in the cell, retinoblastoma is bound to E2F, which is a transcription factor. When retinoblastoma is phosphorylated, it dissociates from E2F, and when it dissociates from E2F, E2F is able to activate, and what E2F does, because it's a transcription factor, is it induces certain genes, and these certain genes are pro-mitotic genes. So these are genes that promote progression through the cell cycle, specifically that promote progression from G1 phase to S phase. So what retinoblastoma is normally doing is essentially binding E2F, inactivating E2F, and therefore preventing the expression of pro-replicative genes, so preventing progression from G1 phase to S phase. Again, once retinoblastoma is phosphorylated, it is not able to inhibit E2F anymore, so it dissociates from E2F, and then E2F is able to fulfill its pro-replicative function. And again, this is just a kind of chart of what's happening, so a flow chart example of the events. So there's pro-replicative signaling upstream. This results in retinoblastoma being phosphorylated, which results in the dissociation of retinoblastoma from E2F. E2F is then able to promote the expression of genes which themselves promote progression into S phase, so promote progression through the cell cycle. And what happens then is the cell successfully progresses through the G1S cell cycle checkpoint. What T antigens can do is essentially prevent this from happening, or not prevent this from happening. They can manipulate this process, so they can nullify the whole process. What these T antigens essentially do is they bind directly to retinoblastoma, and when T antigens are bound to retinoblastoma, retinoblastoma is no longer able to inhibit E2F because the E2F retinoblastoma complex is not able to form. T antigens therefore, by binding to retinoblastoma, promote progression through the cell cycle. So they promote progression from G1 phase to S phase because they are inhibiting retinoblastoma. They are not allowing retinoblastoma to itself control E2F. So therefore, it should be clear that this T antigen, and this is the T antigen of the adenovirus we're talking about here, is inhibiting the activity of retinoblastoma, and in doing so is promoting progression through this G1S checkpoint. And these are some other T antigens that are operating largely in a similar way, though we tend to use the adenovirus T antigen as our example here. And here we have some more examples of the oncoproteins, and specifically the way in which oncoproteins target this particular tumor suppressor protein P53. So P53 is known as the guardian of the genome sometimes, because if there's damage to the genome, or if the genome is being replicated at an inappropriate time, which you often see when you have uncontrolled cell proliferation. So when you have uncontrolled cell proliferation, you have hyperproliferation, the lines between the different phases get a bit blurred, and you start to see genome replication at inappropriate times. And what P53 essentially does in response to the detection of this damage or this inappropriate synthesis is it shuts down the cell and it induces programmed cell death. So it essentially kills the cell. This is a problem from the virus's perspective because the virus doesn't want the cell to be killed. It doesn't want the cell to be shut down. It wants it wants the cell to continue producing new virus particles. So it is therefore attractive from a viral point of view to shut down P53, to target P53. And just like we saw with the T antigen targeting retinoblastoma, here we can see a range of different viral oncoproteins will specifically target P53. Some will target P53 for proteasomal degradation. Now, as I'm sure you know, there are various cellular mechanisms that promote the degradation of specific proteins. That degradation is conducted by proteasomes, and these proteasomes degrade proteins that are tagged with ubiquitin residues. Okay, so certain proteins will be targeted for degradation by the addition of these ubiquitin residues. What viral proteins can do in this case is target P53 for proteasomal degradation by ubiquitinating it. So these viral proteins are essentially tricking the cell, manipulating the cell into destroying its own P53. It's one of the strategies used. Another strategy is similar to the strategy utilized by T antigen to inactivate retinoblastoma. What we have here is adenovirus binding specifically to P53 and inhibiting its activity by binding directly to it. So it's inhibiting the ability of P53 to carry out its normal functions by physically binding directly to it. Finally, viral proteins can sequester P53. This idea of sequestering just means to accumulate it, and by accumulating it, preventing it from fulfilling its normal function in the cell. And this is the third strategy that certain viral proteins can use to inhibit the ability of p53 to exert any kind of control over the cell. Because again, if p53 is not controlled and the cell begins to transform, the role of p53 is to cause cell death, so to induce this programmed cell death to prevent full transformation, to prevent the progression to hyperproliferation and tumorigenesis. So hopefully it's been clear in this last section how oncoproteins, so these are proteins that are encoded by the oncogenes, can actively contribute to transformation. So these can actively induce cellular transformation. Thank you for your time. Quite a complex topic today. Hopefully it all made sense. If anything hasn't made sense, you'll definitely have the opportunity to ask questions about it at the tutorial on Wednesday. So thank you very much. Take care, everyone.